In our previous video, we have discussed that KRAS mutations, uh, KRAS mutations are the one that helps the pancreatic cancer cells in the migrations and metastasis, and whereas the FAS receptors that are been inhibited when the KRAS mutations are on because of the methylations of the promoters in the DNA that helps in the production of FAS receptors. So in this video we will discuss what kind of immune cells that interact with these FAS receptors. As this when the KRAS mutations are turned off, there is an increase in the amount of FAS receptors on the pancreatic cancer surface, and it will interact with the immune cells so that the FAS receptors will trigger apoptosis on the pancreatic cancer cells. In the previous video we have understood how the experimental studies are being done to develop or to prove that hypothesis. In this video we will continue our discussions to go deeper and find out which subpopulation of the immune cells will will interact with these FAS receptors on the pancreatic cancer cells. So this is the experimental procedure that they have used. That is, they took the mouse models in which they inject this PIKP orthopedic mouse models they use, which has where they keep the KRAS mutations on, and after 8 days they introduce this alpha CD4 and alpha CD8 antibodies that helps in the depletion of their respective immune cells that has these receptors. And on the 10th day they have turned off this KRAS mutations by stop this doxycycline treatment, and then they observe the mouse models till its end point or sacrificial points. And this is the panel B to show that these alpha CD4 and alpha CD8 antibodies are working. In the isotope mouse models, they can see both the CD4 plus and CD8 plus populations, and in this alpha CD4 antibodies condition, they can see the no CD4 populations, and in this alpha CD8 antibodies injected mouse models, they see no CD8 immune cells. So this shows that their model works as per their expectations. And thereafter they did the experimental studies or do the statistical analysis in terms of to find out what are the kind of immune cells they can observe in different conditions, or what are the percentage of CD44 plus cells they have find out. And they can see that when the KRAS mutations are on, there is a less number of CD3 plus cells compared to the case when the KRAS mutations are off after 5 days. And when they use this alpha CD4 antibodies, they can see the further increase in this CD3 plus populations. And however, when the CD8 plus cells are depleted, they can see the decrease in the CD3 plus populations. And this is the control, that is CD4 plus and CD8 plus, where they can see that CD4 plus population is completely depleted when this alpha CD4 antibodies are used, and CD8 plus population is completely depleted when this alpha CD8 antibodies are used. And when they compare these conditions with this pancreatic cancer's weight, they can see that when there is a KRAS mutations are on, they can see the increase in the amount of the pancreatic cancer weight and pancreatic cancer tumor weight, and compared to the case when the KRAS mutations are turned on and off, or when these alpha CD4 antibodies are used, and the condition says that KRAS mutations remain on and off. However, when these alpha CD8 populations are reduced and alpha CD8 antibodies are used, then it will cause the increase in the tumor population. This indicates that CD8 plus might plays an important role towards interacting with the FAS receptors. And when they check the survival rate of the mice, they can see that when this alpha CD8 populations, alpha CD8 antibodies are used, they can see the decrease in the survival rate compared to the case when this alpha CD4 populations are used. This shows that the CD8 T cells might play a role towards is interacting with the FAS receptors compared to the other subpopulations of the immune cells. Next they did the histocompatibility analysis of the tumors at different conditions, and they can see that the the tumors condition in the isotype is remain similar to the case when this CD8 alpha CD8 antibodies are used. So this further indicates that CD8 plus cells has an important role. However, when this isotope are on and off, and if this condition is similar to this when the alpha CD4 antibodies are used. So this shows the CD4 antibodies might have low roles, and these things they have quantified using this computational statistical analysis, and they can see the similar things, that is when the relative percentage of this tissue phenotype, that is in the pancreatic cancer cells, is high in the case of when this isotype is on and when this alpha CD8 antibodies are used, compared to the case when these isotypes are on and off and this alpha CD4 antibodies are used. So this also supports the point that CD8 plus cells might have an important role, and depletion of CD8 plus cells will cause increase in the pancreatic tumor cells or pancreatic tumor weights. Next they did the analysis of the clinical data of this murine samples, where they identified different cell populations that are found out in the murine that is infected with this PDAC, and out of this they further classify these T cells to find out what are the different kind of T cells that are, what are the different populations of the T cells that are present in these samples, as they want to find out which subpopulations are related with these FAS receptors in the clinical samples. And in this they are able to find 5 different clusters of the T cells that can be seen here, 1 2 3 4 5, or they have labeled it as 0 1 2 3 4, and in this subpopulation and within this T cells they also detect, or they also try to identify what are the different kind of the CD3 plus CD4 plus CD8 plus and FAS receptors are present. Not within these T cells, but within these murine mouse models, they check what are the different kind of T cells and FAS receptors are present, and they can see that the CD8 plus cells seems to be in line with FAS cell receptors. And later on they check in the different subpopulations T cells metaclusters that what are the expressions levels of CD8 plus and CD FAS cell receptors, and they can check that in the these FAS cell receptors are high and in line with this CD8 plus expressions in the fourth cluster. However, we can see they can also see some CD8 plus expressions in the cluster 0. However, there is no FAS cell receptors, but this fourth clusters is in line with the previous observations that CD8 plus cells might interacting with these FAS cell receptors. Later on they check the methylation of these FAS promoters at different conditions. So this is slightly different from what we have discussed so far. Here they are checking the FAS receptors. What are the reasons these FAS receptors are not, what are the reasons they are checking the differences in different conditions that FAS receptors, why the FAS receptors are produced low. So the reasons that they are targeting is this methylation of these FAS promoters. So when the conditions are on, they can see that high methylations of these FAS promoters. So this will less leads to the transcription of these FAS receptors, compared to the case when this after 3rd days this FAS receptors has been turned off, and when it's 5 days when it is turned off, then we can see that less methylations, and this indicates that KRAS mutations controls or helps in the methylations of this FAS promoter. Thereafter they check FAS cells at different conditions with, that is with DNA methylation inhibitors, and when the KRAS mutations are on, they check the FAS receptors when with DNA methylation inhibitor. So when these FAS KRAS mutations are on, they see negligible amount of DNA methylations compared to the case with these inhibitors. They can see the increase in the amount of FAS receptors. And however, when the KRAS mutations are off, they can significantly see the increase in the FAS mutations. And when the KRAS mutations off with DNA inhibitor, there is not much change. So this further indicates that there is not much increase. There is not in any different factors that will cause the increase in the FAS receptors formations. However, this indicates that there could be another factors that will also contribute towards this FAS receptors methylations or FAS receptors less productions. And later on they also check this 5-day off and 5-day on conditions without any DNA inhibitors, and 5-day off and 5-day on conditions with DNA inhibitors. So here they also see the increase in this FAS receptors. So this made from this data they made a point that it's the DNA methylations of these FAS promoters that is affecting the FAS receptors formations on the pancreatic cancer cell surface. And later on they check different factors that cause the epigenetic changes within the tumor cells. So one of the factors they use is, one of the proteins expression levels that they use is DNMT1, and the other one is S100Z. And they can see that high expression levels of this epigenetic these proteins that are expressed during epigenetic changes are during this on and off conditions. So however, they are low when these KRAS mutations are turned off. So this indicates that there is epigenetic changes that is going on within the pancreatic cancer cells when the KRAS mutations are on, off, and on, or when the KRAS mutations are on, compared to the case when these KRAS mutations are off. And therefore they check their relative enrichment of this FAS receptors with this different relative expression of the FAS receptors with these different epigenetic related proteins. And they can see that high expression of these FAS receptors when this H3-2K7 proteins are expressed. So they can see when the KRAS mutations are turned off, the higher the relative expression of these proteins when this H3K27AC proteins are expressed or highly expressed. So from this they made a schematic overview, like what is happening when the KRAS mutations are off and on. The KRAS mutations are on, then these two proteins might be preventing the transcription of these FAS receptors, and the transcriptions is suppressed. However, when the KRAS mutations are turned off, then these two proteins, that is EZH2 and DNMT1s, are not restricting the promoters of these FAS receptors, and the FAS receptors can be translated. As from this data that is in panel G, we can see that when this with the expression of these proteins, they can see the less expression with the expression of this DNMT1 and EZH2 proteins. We can see the less expression of these FAS receptors compared less than these normal levels. And so however, when these KRAS mutations are turned off, they can see the high expression of this. They can see when the KRAS mutations are turned off, they can see the low expression of these epigenetic factors. That is, those epigenetic factors is not restricting the promoters of these FAS receptors. Therefore, the transcription of these FAS receptors is been enhanced. And same things that they have observed in the immunostainings. That is, when the KRAS mutations are turned off, they can see the high expression of these FAS receptors. At the same time, this caspase 3 that is responsible for, that is one of the proteins that is triggered during the apoptosis is triggered. So that can also be seen when the KRAS mutations are turned off. However, there are very low amount of this FAS receptors can be seen when the KRAS mutations are turned on. And similarly, they quantify this analysis in this statistical form, and they can see that the KRAS mutations are off. They can see the increase in the number of these caspase 3 proteins formations, or the CK19's FAS receptors caspase 3 formations. High amount of these FAS positive and caspase FAS receptors and caspase 3 formations compared to the case when the KRAS mutations are turned on, or when the KRAS mutations are on. So these all experimental results indicates that it's the CD8 plus cells that contributes or that interacts with the FAS receptors on the pancreatic tumor cells to trigger apoptosis. So I hope you got to understand how these experimental data communicate us to this result. If you need to understand more about these experimental studies in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.